Hello, everybody. Today we're going to talk about one of the biggest discoveries in biology, how scientists figured out that DNA carries hereditary information. That might sound obvious to us today because DNA is everywhere in modern biology, medicine, forensics, TV, etc. But for a long time, scientists had no idea what molecule controlled heredity. In this lecture, we'll follow the major discoveries that connected DNA to genes and heredity, and we'll also see how bacteria and viruses became incredibly important tools for understanding genetics, or at least we'll briefly touch on that. By the end of this module, you should be able to describe the discovery of nucleic acids and nucleotides, explain the key experiments that characterize DNA, describe how microorganisms help scientists uncover gene function, and explain how scientists establish that DNA is the hereditary material. All right. So you can see examples of inherited traits in this picture alone, right? We've got the father and the child, and they share some characteristics. Features are passed from parents to offspring. Today, we know that DNA controls those traits, but scientists in the early 1900s were not sure what molecule carried hereditary information. In fact, many scientists thought proteins were the genetic material because proteins are chemically very complicated. DNA seemed too simple to store all the information needed to build a living organism. So the question became, what molecule actually stores hereditary information? In 1869, a Swiss scientist named Friedrich Miescher isolated a strange substance from the nuclei of white blood cells. He noticed this material contained large amounts of phosphorus, which was unusual because it came from the nucleus. He called it nuclein. We now know this material was DNA. At the time, though, nobody understood what nuclein actually did. Scientists had discovered DNA long before they actually understood what its importance was. Now, scientists later discovered that DNA is built from repeating units called nucleotides. Each nucleotide has three parts: a phosphate group, a sugar, and a nitrogenous base. The four bases in DNA are adenine, thymine, cytosine, and guanine. These repeating nucleotides form long chains that we can call polymers that make up DNA molecules. Even after scientists understood DNA's chemical structure at the basic level, many still believe DNA was too repetitive and simple to carry the genetic information. So Frederick Griffith was studying bacteria that cause pneumonia. He worked with two strains of Streptococcus pneumoniae, the smooth strain or the S strain, which was deadly because it had a protective capsule. We talked about capsules before, and they can actually protect the cell from the immune response. And the rough strain or the R strain, which was harmless. Now notice the images on this screen. It's showing the different outcomes to the mouse. When Griffith injected live smooth bacteria or S bacteria into the mice died. When he injected live R bacteria, the mice survived. But here's the part that was pretty cool and surprising. When he mixed heat-killed S bacteria with live R bacteria, the mice still died. So even more importantly, live S bacteria could be recovered from the dead mice. So if we remember back to Koch's postulate, we've kind of got something going on here. So we know that something from the dead S cells transformed those harmless R cells into deadly S cells, right? So this process of transforming became known as transformation. But again, at this point, we don't know what that molecule is. We just know that the dead S cells can give something to the living R cells to make the living R cells become deadly, right? So that's kind of a weird thing, especially to Griffith back when this experiment took place. Now, in this experiment by Avery, McLeod, and McCarty, they were able to destroy different parts of the material, right? So they could either destroy proteins, destroy RNA, or destroy DNA, right? And so Avery and his colleagues systematically destroyed these different molecules in bacterial extract. So he's kind of working off of Griffith's idea. And when they destroyed proteins, transformation still happened. When they destroyed RNA, transformation still happened. But when they destroyed DNA, transformation stopped. Now, as you might imagine, this was very exciting to them. This gave strong evidence that DNA and not protein was responsible for heredity. Even then, many scientists still resisted the idea because protein seemed much more complex. And these simple, DNA molecules did not seem like they could actually do the job. Now, then come along Hershey and Chase. Alfred Hershey and Martha Chase used viruses that infected bacteria. We call these viruses bacteriophages. They'll come up again later. These viruses are simple. They're basically made of DNA surrounded by protein. That simplicity made them perfect experimental tools, though. The researchers labeled viral DNA with radioactive phosphorus and labeled protein with radioactive sulfur. All right. And so when the viruses infected bacteria, the radioactive DNA entered the bacterial cells, but the protein stayed on the outside. The image on this slide helps visualize this process. And this experiment provided very strong evidence that DNA carried hereditary information because when they took the infected bacterial cells, they found that radioactive phosphorus inside of the cell and not the radioactive sulfur that was in the protein coating. All right. And so this, kind of jumps us ahead a little bit, but microorganisms became incredibly important for genetic research because they grow quickly, they reproduce rapidly, and are easy to manipulate experimentally. Scientists could observe genetic changes much faster in microbes than in plants or animals. And so Beadle and Tatum, George Beadle and Edward Tatum, used bread mold to study mutations in metabolism. Their experiments showed that genes control the production of enzymes. This led to the one gene, one enzyme hypothesis, which connect genes directly to biochemistry. The idea here is that there is a portion of the DNA that codes for a gene. That gene codes for an individual protein. Now, using X-ray diffraction data produced by Rosalind Franklin, Watson and Crick proposed the double helix model of DNA in 1953. The structure explained how DNA could store information, replicate itself, and pass hereditary information to future generations. So in this, they have complementary base pairing, where A's bind the T's and C's bind the G's. And that pairing mechanism we now know is critical because it allows DNA to be copied accurately during replication. And so to kind of bring this all together, let's look at the big picture. DNA was first discovered as this mysterious substance called nuclein. Scientists later discovered DNA is made of nucleotides. Experiments with bacteria and viruses demonstrated that DNA carries hereditary information. So, like, we owe our modern understanding of genetics largely to microorganisms. Microorganisms helped scientists connect genes to cellular biochemistry, and finally, the discovery of the DNA double helix explained how genetic information is stored and copied. Together, these discoveries form the foundation of modern genetics and molecular biology. And in the next couple of lectures, we're going to continue to introduce these general ideas. And starting next week, we'll go deeper into understanding how how this material helps genetic information get passed along. And with that, thank you for watching this lecture and talk to you soon.